Thank you. Thank you. So histone marks control many aspects of the genome regulation. We know that they mark positions like promoters, enhancers, and exons. They also influence the structure of the chromatin. We know this mainly through ChIP-seq, which use sonication to disrupt the DNA sequence, and then through immunoprecipitation, we filter out those histone marks of interest. However, this produces a footprint that can be several times the length of the mononucleosomal DNA. In practice, this means that when we trace back these footprints, we are doing it over a genomic window, not a particular nucleosome. Why this is important? Because there are some elements, like promoters, where there is information that different histone marks are co-localized in the same window. So questions like, are these different histone marks co-localized in the same nucleosome or in neighboring nucleosomes cannot be addressed by the regular ChIP-seq protocols. However, this can be overcome replacing the sonication step by enzymatic digestion. Provided enough digestion time, we can digest all the linker DNA up to the point to generate mononucleosomal footprints. Still, there are some technical difficulties to apply this. So here we develop computational and statistical method to take the power of enzyme-based ChIP-seq protocols to trace this histone mark to individual nucleosomes. In particular, we use MNase digestion of DNA coupled with ChIP-seq. This data was generated by Dr. Xu Xiao from Song Lab at UCSD. And he created libraries of 4 different histone marks on mouse embryonic stem cells. So before we trace these histone marks to individual nucleosomes, we need to know where are the nucleosomes. So we use our own MNase ChIP-seq libraries, and we combine it to call the nucleosome positions. For this, we first verify that we are generating mononucleosomal footprints. So here you have a triga-14 methylation, both replicates. This is a histogram of the fragment sizes distribution, which go from 0 to 300, and here you have 150. So most of these reads are actually mononucleosomal fragments. The same goes for all the 4 different histone marks. We also supplemented our MNase ChIP-seq libraries with MNase-seq data from Carone et al. This is a publicly available data set. However, in their protocol, they were interested also in determining the position of molecules smaller than nucleosomes, like transcription factors. So they have a heavy tail in this side. So this author discarded everything that is below 135 bases or above 155. We did the same, so we only used the 90 million reads in this region, in the mononucleosomal region. We then pulled this MNase-seq data along with our MNase ChIP-seq data to generate a database of 400 million reads, which we then input in a software called IMPS to determine what are the nucleosome positions. We found around 10.2 million nucleosomes. These roughly correspond to 84% of the expected nucleosomes in the mouse genome. We know this just by dividing the length of the mouse genome by the mononucleosomal fragment size plus the linker sequence, which is typically found between adjacent nucleosomes. Here I'm just showing an example of a region where the pink peaks correspond to the pooled datasite distribution, and the gray bars correspond to the nucleosomes. So you can see that most of these positions are very well defined, and they are not merged with neighboring nucleosomes. So the next step was, now that we have the map of these nucleosomes, we want to determine which one of them are actually marked by a particular histone mark. So we developed a procedure where, knowing the location of each nucleosome, we use as an index of enrichment the counts of histone footprints on each one of them. As you may know, there is a confounding effect between the sampling bias of the MNase-seq data and the actual footprints of the histone marks. So to address this problem, we stratify all the nucleosomes according to the number of MNase-seq reads that they have. So for example, here we took all the nucleosomes that contain 7 MNase-seq reads, and then we compute the expected value of the histone footprints, as well as the 95% quantile. Why? I'm going to explain in a second. So here we have for all 4 histone marks the distribution of the count of histone footprints in the y-axis against the count of MNA-seq footprints. And as you can see, the larger the amount of MNA-seq reads, the larger the expected amount of histone footprints. Therefore, for each one of these values, the blue line corresponds to the expected value, we co-enrich only those nucleosomes that are above the 95% quantile. So for example, for a nucleosome that contains 15 MNA-seq reads, we call it enriched by H3K9 methylation only if it contains 6 or more footprints of that histone mark. These generated between 600,000 up to 800,000 enriched nucleosomes across all these 4 histone marks. So now that we have the nucleosome map and we know which one of them are marked by a particular histone, we ask the question, do these histone marks tend to be co-localized or independently distributed in the genome? So what you are seeing here is just the number of observed combinatorial patterns. So we took all 16 possible combinations of these 4 histone marks. For example, here, nucleosome that only contain H3K27 methylation. Here, nucleosome that contain all 4, and anything in between. We found that most of the nucleosomes tend to gather only one histone mark, especially H3K9 methylation, but there were significant amount of nucleosomes having at least 2 histone marks. We use a chi-square test to compute the significance of this against the null hypothesis that each one of these histone marks is independently distributed in the genome. And we found that all these counts were actually significant, meaning that the null hypothesis is unlikely to account for this account. To pinpoint the co-localizations more important among these data, we define a co-localization score, which is only the observed co-localization count, this is these bars, divided by the expected co-localization count under the null hypothesis. And then we just apply, we just log transform this data. And we found that the most enriched co-localization marks are the ones that contain at least H3K27 methylation, along with H3K9 methylation, H3K27 acetylation, and H3K4 methylation. This was at the beginning surprising because H3K4 methylation has been linked to active expression, whereas H3K27 methylation has the opposite role. However, in embryonic stem cells, there is known that there are bivalent domains. That is, regions of the genome that contain these opposite histone marks. So our next question was, are these most likely the result of bivalent domains? So we explored the distribution of bivalent domains defined using typical ChIP-seq data. So here in this track, you can observe H3K4 methylation using sonication, followed by ChIP-seq, and the same for H3K27 methylation. So this region, having the repressive mark and the activator mark, is called traditionally a bivalent domain. And then we count how many of those contain what we call a bivalent nucleosome. For example, this one, where we have a nucleosome that contains both marks at the same time. Out of 400 bivalent domains, which go between around 1,000 bases up to 6,000, with a typical value of 2,000 bases in length, we found that around 26% of them contain at least one bivalent nucleosome. We asked if this intersection is significant or not. To test this, we sampled the genome 1,000 times. For each sample, we generated a distribution of bivalent domains randomly picked on the genome, having the same distribution as the observed bivalent domains. And for each one of those iterations, we counted the proportion of bivalent domains that contain at least one bivalent nucleosome. The blue distribution corresponds to this background, and here, the peak corresponds to the 26%. And as you can see, this background distribution is unlikely to express that level of intersection. So based on this, we conclude that these bivalent domains are actually enriched by nucleosomes having both histone marks co-localized. Another functional element that we studied was the transcription starting site. So here, again, you have the different combinatorial patterns, and here you have all the genes, regardless of gene expression, centered around the transcription starting site, plus minus 1.6 million bases. And you can see that almost all the combinatorial patterns are enriched at around the transcription starting site, but they fade away as we move downstream or upstream. This leads us to speculate that these nucleosomes contain information that is important for the regulation of gene expression. We further test this hypothesis by now dividing the genes according to their gene expression. So we use publicly available RNA-seq data, and we divide the genes between silent and highly expressed. Among the silent ones, this one, you can see that the combinatorial patterns are almost equally expressed around the transcription starting site, also showing the position effect. Interestingly, among the highly active genes, we found enriched only those that contain the previously known activator marks, H3K4 methylation and H3K27 acetylation, whereas those nucleosomes that contain the repressive mark are depleted. So this makes sense with prior knowledge. What is interesting of this figure is that still there are some nucleosomes that contain the repressive mark and are enriched. For example, this one here, which contain H3K9 methylation, a repressor of gene expression. However, all those that contain the repressive mark are also co-localized with H3K27 acetylation, but not H3K4 methylation. So we deduce that the activator role of this mark overpowered the repressive role of the others, but not H3K4 methylation, which is superseded by the repressive mark. So we also look into more detail into the transcription starting site. This time we use the MNA-seq data to call the nucleosome positions of what is called canonical nucleosomes. So here, based on the MNA-seq data, you observe a deep valley. This is expected. The region upstream of the transcription starting site is typically depleted of nucleosomes for the transcriptional machinery to recognize the sequence that are there. And downstream, you have a strong peak. This is called the +1 nucleosome. And based on these peaks, we define from the -5 up to the +5 canonical nucleosome. And just using the aggregated enrichment across several genes, we found that H3K4 methylation and H3K27 acetylation are mainly enriched at the +1 nucleosome, with highly active genes in all colors but red, having stronger signals than the silent genes. H3K9 methylation also show a little bit of enrichment, but not as strong as the previous two. However, this was surprising. We have that the repressive mark also is enriched among the active genes compared to the silent ones. So we deduce that this is a result of the previous slide, where we observed this repressive mark only if we have the activator marks in the same nucleosome. So I'm going to jump some results. I want to thank my advisor, my co-authors, and my sponsors. Please, if you have any question.